All right. So my presentation in final project is on Gertrude or Trudy Elion and the immunosuppressant drug azathioprine. So as some background on Trudy, she was born in 1918 and graduated high school at 15 years old. And this was the same year when her grandfather died of stomach cancer. And this really inspired her to pursue a career in science and medicine. And so because she had really good grades in high school, she was allowed to attend Hunter College for free. And she graduated from there in 1937, summa cum laude, with a degree in chemistry. However, despite her degree, she wasn't able to find a paying research job due to her gender. And she also wasn't able to get any graduate research scholarships. And she couldn't afford to go to graduate school without a scholarship. So instead, she had to work as a secretary and a high school teacher to earn money. She did attend NYU and earned a master's in 1941 in chemistry, but even after that, she still couldn't get a graduate research position. And so she was eventually able to get a chemistry-based job working as a food quality supervisor, testing the acidity of pickles, which is chemistry related, but wasn't really the field that she was hoping for. So she did finally get an actual position at the Burroughs Wellcome Pharmaceutical Company, working as an assistant to the researcher George Hitchings. During this time, she also began working on graduate studies at Brooklyn Polytechnic Institute, which is now NYU's Tannen School of Engineering. However, she was doing this part-time and the school really wanted her to do full-time, and she didn't want to give up her job to do that, and so she couldn't finish her graduate studies. In the later parts of her career, she became an adjunct professor at Duke University from 1971 to 1983, which is the year when she retired from her job at the pharmaceutical company. And then she became a research professor at Duke until she died in 1999. Though she never got any official PhD, she did earn an honorary PhD from NYU's Tannen School in 1989, as well as an honorary SD from Harvard in 1998, and many other honorary degrees from other institutions. So a bit about the work that she did with George Hitchings and his goal of rational drug design. So this design method is similar to what we learned of target-oriented drug development, where you use what you know about the biological target in order to develop a better drug. So what he was trying to do was to develop antagonistic nucleic acid derivatives, molecules that would trick cancer cells into using these artificial compounds for growth. And since these artificial compounds are not actually nucleic acids, they wouldn't be able to grow as well as with the regular acids. And this would also allow them to be targeted and selectively destroyed without harming your regular cells. So what Trudy worked on was she synthesized many antimetabolites of purines, and from this research, she helped develop many anti-cancer and other forms of drugs. So as some background, purines and pyrimidines are basic building blocks of DNA and RNA, and they're really important for this. And so purines, as shown in the one on the top, are heterocyclic aromatic rings, and it's a pyrimidine fused to imidazole. These are the DNA RNA bases adenine and guanine. They are purines. Then pyrimidines is just a single heterocyclic aromatic ring. This is the one on the bottom, and these are in the DNA and RNA bases cytosine, thymine, and uracil. So her first breakthrough was mercaptopurine 6-MP. This is the figure shown on the right. It's just a basic purine with a sulfur instead of a nitrogen or an oxygen at that position. This drug is also known as purine-thol, and it was FDA approved in 1953 as the first treatment for leukemia. And it's still widely used today as treatment for leukemia, especially in children. And it is on the World Health Organization's list of essential medicines. So getting into azathioprine, also known as Imuran, this is the drug I'll be talking about more. This is shown on the right. So it's basically just the 6-MP with an imidazole ring attached on top, a substituted one. And azathioprine is an immunosuppressant drug, and it was FDA approved in 1968. It's used to treat rheumatoid arthritis, Crohn's disease, and most notably is used in organ transplants to help prevent rejection. So because it suppresses your immune system, you won't have an immune reaction to the foreign organ, and it will be taken more easily. And this drug is also on the World Health Organization's list of essential medicines. And in 2018, it was the 358th most prescribed medicine in the United States. So even though this drug was developed a long time ago, it's still very impactful. So as for the synthesis of azathioprine, luckily for her, this precursor imidazole ring had already been synthesized. So she didn't have to make this, she could just get it. And then she fused that to this reduced form of the 6-MP to form the azathioprine. So it was a very simple synthesis for them to do. The mechanism of azathioprine is, so azathioprine is a prodrug, and it's just converted to the 6-MP in the body. The attached imidazole ring helps it improve its bioavailability and target the right spots. And what the 6-MP does is that it gets incorporated in DNA synthesis. And since it's not a normal purine, it forms non-functional DNA, and that stops replication from occurring. And that can help inhibit replication of cells that you don't want, like cancer cells. What azathioprine and the 6-MP drug does, it inhibits the glutamine phosphoribosyl pyrophosphate aminotransferase enzyme, which GPAT, which is involved in purine biosynthesis. And so this prevents regular purines from being formed. And so this works through a pathway called product inhibition. So 6-MP looks a lot like the product that the enzyme is intending to create. And so it stops it from making more because this enzyme thinks there's already enough in the cell. And this means that there's only 6-MP and not the regular purines for DNA synthesis. And so it can't form functional DNA at all. And this is, of course, most active on cells that are actively replicating. So this includes cancer cells, but also immune cells. And so it's very effective as an immunosuppressant. Some other drugs that are that she helped develop, these are all also on the World Health Organization's list of essential medicine. This includes zidovudine, or azothymidine. This is shown on the right. It was FDA approved in 1987, and it's the first treatment for HIV and AIDS, and is also preventative. And you can see here that it doesn't have a purine, but it does have a pyrimidine up here. And so purines and pyrimidines were very prominent in the drugs that she helped to develop. Yeah, and she's also developed a lot of other drugs that aren't on this list, like many other drugs, some of which are also on the World Health Organization's list, but some of which aren't. But yeah, and during her career, she earned lots of awards and honors. Most notably, she received the Nobel Prize in Physiology or Medicine alongside George Hitchings and another scientist, Sir James Black, for their discoveries of new principles of drug treatment. So this is the rational drug design that they were doing in 1998. She was also elected of members of lots of academies, and she received lots and medals of science and honor. And also notably, she was the first woman inducted into the National Inventors Hall of Fame in 1991. The first of many, of course. Yeah, and so here are my references. Thank you for listening.